Hello, everybody. In this lecture, we're going to talk about antibiotic resistance. So let's get right into it. By the end of this module, you should be able to explain what is meant by antimicrobial resistance and compare to both intrinsic and acquired resistances. Name and describe the three categories of acquired antimicrobial resistance tools that microbes may use to thwart drug action, and discuss how to use the use of antimicrobials selects for resistant microbes. All right, so um, when we're thinking about antimicrobial resistance, this is going to occur when a microbe is not affected by a drug. All right, they're not inhibited or they're not eliminated. And when you have resistance, some people can refer to antibiotic resistant organisms as superbugs, although I don't. And you may also hear the term superinfection, which means that you have the only thing causing the infection is superbugs. Now, this down here shows you this idea that you can have resistant organisms. Susceptible organisms die when they receive the treatment, and then those resistant organisms thrive and grow in abundance. And then they may also transfer that resistance to other organisms. Now, we have two types of resistances. We have intrinsic and acquired resistance. And so an intrinsic resistance is going to be something that the cell has naturally that helps produce or prevent infection, or sorry, prevent effects of the drug, such as the type of cell wall, the type of membrane, the ability to form endospores, etc. Acquired resistance is going to be something where you either get a mutation or you receive a gene through horizontal gene transfer that that gives you resistance to that drug. And so, again, our intrinsic resistances are natural resistance to those drugs. This can make certain organisms hard to eliminate. One example is that waxy mycolic acid layer that we talked about that TB has, and that helps prevent many drugs from entering into the cell. Gram-negative bacteria have an outer membrane. Microbes produce biofilms that can be difficult to diffuse through as well. So these are all intrinsic resistances. Acquired resistance, on the other hand, is going to come from some sort of genetic mutation, and that's going to be through something like conjugation, transformation, or transduction. Now, when a microbe is avoiding a drug, there are three primary ways of doing that. They can alter the target of the drug, they can inactivate the drug, or they can reduce the concentration of the drug inside of the cell. So here's a little image showing that. Here, you change the target to where this won't fit inside of the active site anymore. Down here, we have drug inactivation where we break it down. And here we're effluxing out, or we're preventing permeability, or reducing permeability so that drugs can't pass through into the cell. Now we can do target alterations. So one way that this happens is you can actually change the genomic code. This can happen through mutation. In this case, we've got an example of rifampin resistance. And here, all of these are different amino acid changes. Like you can see that this TTC coded for phenylalanine. There's a mutation to TTA, gives you a leucine. So all these different mutations have changed the amino acid code of the RNA polymerase that rifampin actually affects, but it doesn't affect the RNA polymerase's ability to function. You can have drug inactivation. This happens oftentimes with beta-lactams, like penicillin. And so beta-lactamases will actually cut this beta-lactam ring, and that'll result in an inactive form of the drug. Another example would be adding chemical groups. So down here, we actually add an acetyl group to chloramphenicol to inactivate the chloramphenicol. These the ability to do these things can oftentimes be carried on plasmids. Um, and those plasmids can go from organism to organism through the different methods of horizontal gene transfer. Now, you can always lower the concentration of the drug. This can be a very effective way of limiting the amount of drug um, entering the cell. We can do that through actively effluxing the stuff out of the cell, or by limiting uh, permeability. So one example here is that fluoroquinolones and carbapenems require porins to enter into the cell. Many drugs do need some sort of help getting in. And so if the bacteria adjust their porins, if they have some sort of mutation that changes the porin shape, it can actually reduce the amount of drug that enters the cell. Um, Now, this can also happen by just having more pumps. If you have more efflux pumps, you can pump more stuff out. Um, you can also get these either through mutations that cause overexpression or through horizontal gene transfer. Now, one thing to keep in mind is that our impact on antibiotic resistance is very important. We do that through prescribing drugs, through animals, because a lot of times livestock receives drugs, etc. And so many patients don't actually finish their drug regimes. This can actually lead to pathogens being exposed to subinhibitory, like below the minimum inhibitory concentration. That's what MIC is. And then they can start to develop resistance. And we oftentimes give drugs to people who don't need them. And in some countries, there's no real regulation, right? So this could be because this can lead to higher prescriptions to people who don't need it. You can also get this through misdiagnosis, like if someone has a viral infection, an antibiotic's not going to do any good. Patients sometimes pressure their clinicians to give them drugs as well. I want to show you this video if I can get it to play. It's pretty awesome. Okay. I'm going to load the video because let me see if I stop it, if I can get it to play. Nope, didn't work. All right. Well, I apologize. I'm going to post the video on there. You should be able to explain antibiotic resistance, compare intrinsic and acquired, and you should be able to describe 3 categories of acquired antimicrobial resistance, how you get them, and how use of antimicrobials can actually lead to antibiotic resistance. To answer that last one, the video that I'm going to post is going to be very helpful. All right. Thank you all, and I'll talk to you soon.